So the next awardee is a winner of the MDS Foundation Young Investigator Award, and is Dr. Supak from the Bijar Lab in UC San Diego.
Thank you very much for this award. I'd like to tell you all about an upcoming project that I'll be working on. So research in clonal hematopoiesis and our understanding of it has grown significantly over the past 5 years since CHIP's association with age and adverse outcomes was first described. We know CHIP represents a precursor state and is also an important risk factor for cardiovascular events. Clonal hematopoiesis also occurs in other settings, such as after receiving chemotherapy for other cancers, and in this context, frequently harbors mutations in epigenetic modifiers, spliceosome genes, myeloid transcription factors, and is also enriched for mutations in TP53, increasing the risk of developing therapy-related disease. In this case control study published by the Moffitt Group found that the presence of a detectable myeloid clone after chemotherapy increased the likelihood of developing a therapy-related myeloid neoplasm. As you can see in the bar graph, TP53 was among the most commonly mutated genes in cases, and in the plots on the right and in the fish plot, you can see that therapy-related disease was often driven by expansion of pre-existing TP53 mutant clones, as depicted by the red lines in the plots on the right and the gray lines in the fish plot. And these clones are thought to arise under the selective pressures of chemotherapy. A similar study was also published by the MD Anderson group as well. But however, the presence of somatic driver mutations alone does not seem sufficient to induce clonal expansion, and this is mainly based on the observation that the size of CHIP clones can remain stable at low levels for many years. In this study by the WashU group, the researchers looked for clonal hematopoiesis in longitudinally banked blood samples from the Nurses' Health Study, using error-corrected sequencing to facilitate detection of rare variants below the error rate of standard next-generation sequencing. They found clonal hematopoiesis in 95% of the studied individuals. The mutation patterns were those associated with aging, and as you can see in this figure, the clones were very stable over time, although more occurred with age. And about a quarter of the mutations were persistent at 2 time points a decade apart. So presumably, some selective pressure is needed for clonal progression, since we know that the majority of individuals with CHIP do not develop a hematologic malignancy. Additional cell extrinsic factors, such as inflammation, likely cooperate with gene mutations to contribute to clonal progression and disease development. As we have heard from many great researchers here, external selective pressures like inflammation can further shape the clonal architecture. Work from the Moffitt Group has shown how malignant clones can direct pro-inflammatory circuits through the NLRP3 inflammasome, which is shown by the wheel-looking figure in the middle of the slide, to drive beta-catenin activation and subsequent propagation of mutant clones. Inflammasome activation actually begins with NF-kappa B-induced transcriptional priming of inflammasome proteins and pro-inflammatory cytokines, as you can see toward the bottom of the slide, to facilitate inflammasome assembly and an inflammatory microenvironment. So somatic mutations actually can activate NF-kappa B and trigger ROS generation, which activates both the inflammasome assembly and beta-catenin signaling, and also primes cells to undergo pyroptosis, which is inflammatory cell death that is mediated by caspase-1, to ultimately support an inflammatory microenvironment and to support mutant clone self-renewal and proliferation. This circuit creates a positive feedback loop that reinforces inflammasome activation. And so we ask, can we target inflammation to reduce clonal burden? So metformin is a widely available diabetes drug with a well-known safety profile and has been studied in several cancer prevention trials. Metformin exhibits anti-inflammatory activity independent from its anti-glycemic effects and suppresses the inflammatory response by modulating oxidative stress and inhibiting NF-kappa B through AMP-K dependent and independent pathways. AMK activation plays a crucial role in the regulation of inflammatory signaling. We observed that following metformin treatment of PBMCs from patients with MDS, the expression levels of phosphorylated AMK were upregulated compared to no treatment, showing that metformin can inhibit inflammatory pathways that are dependent on AMK in primary MDS cells. Anti-neoplastic effects of metformin have also been described for several other hematologic malignancies. And on the right side of the slide, you can see that metformin treatment was able to significantly reduce cell proliferation and clonogenicity in JAK2 mutant MPN cell lines. So the incidence of therapy-related disease is expected to rise in parallel with the increase in cancer survivors and with the expanded use of known leukemogenic agents, highlighting the need for preventative approaches to reduce the risk of therapy-related disease in individuals who are otherwise cured from their primary malignancy. While we cannot reverse genetic mutations, the question now is whether therapy-related clonal hematopoiesis can be modified by targeting inflammatory signaling pathways to alter the natural history of malignant transformation. So we will plan to study the preferential anti-inflammatory effect of metformin on clonal behavior by leveraging banked patient samples from the Reach for Health trial. The Reach for Health trial was a large, randomized, placebo-controlled trial that investigated the impact of metformin on inflammatory prognostic biomarkers in 333 breast cancer survivors. In this trial, metformin was able to lower CRP levels and may also slow or halt expansion of mutant clones associated with therapy-related clonal hematopoiesis. So we plan to combine error-corrected sequencing with targeted capture to study blood samples before and after 6 months of daily metformin from breast cancer survivors who received prior adjuvant chemotherapy for locally advanced breast cancer. And these women represent a high-priority population likely to harbor TP53 mutations. We will also sequence patients, breast cancer survivors who did not receive prior chemotherapy as a comparator group for the impact of chemotherapy on clonal hematopoiesis, mutation type, and clonal burden at baseline. And we will also examine our findings against clinical endpoints, including the development of hematologic malignancies, the development of hematologic abnormalities, cardiovascular events, event-free survival, and overall survival. So my talk was pretty short. Hopefully kept everyone on time, but I look forward to this project, and I am grateful to the MDS Foundation for their support, and I'd like to thank everyone on this slide. Thanks.
Thank you very much for a